Hello everyone, welcome to BioExplaining. In this video, we are going to talk about the topic of sexual reproduction in flowering plants. This topic comes under the unit of reproduction and the lecture is mainly going to be focused on the NCERT Class 12 Biology textbook. So yes, let us begin. So we all know that flowering plants are known as angiosperms. But much before the flower is actually present on the plant, there are various other activities that take place. So all of these activities are together termed under pre-fertilization events, right? So what we have said that much before the actual flower is seen on the plant has already decided that it's going to flower. So what are those different decisions that we are talking about? There are various kind of hormonal changes. There are various kind of structural changes that occur. So these hormonal and structural changes are initiated, which lead to differentiation. And once differentiation happens, it further leads to development of the floral primordium. So what is this floral primordium? This floral primordium means that there are buds created at the end of the stems. So we know that there are two kinds of buds that can be present. One is at the end of the stem, the terminal part, which is the apical bud, and the other is the lateral buds. Okay, so inflorescences are formed. What do we understand by the term inflorescence? Inflorescence refers to the arrangement of a cluster of flowers on the floral axis. So from this inflorescence, floral buds are formed, and then from the floral buds, an entire flower is formed. The flower contains the male and the female reproductive part, which is androecium is the male part and the gynoecium is the female part. Okay, so just a very quick suggestion. If you want to revise the structure of a bisexual flower, what are the four different whorls present, what is their function, then please consider going back to my video on the structure of a bisexual flower. And once you're done with your basics, you're good to go. Get back to this video and let's continue. Okay, so let us start discussing about the androecium, that is the male part of the flower, and the individual units are known as the stamens. If you look into this diagram very carefully, you can note that there are two parts to the stamen. One is the long and the slender stalk that we are terming as the filament, and at the tip there is the bilobed structure which is known as the anther, right? The end part of the filament, that is the proximal part, is either attached to the thalamus or it is attached to the petals of the flower, right? And the length of the stamens and the number of stamens, this is not fixed for every flower. Okay, so the length and number of stamens, this can differ with every species that we have. Fine. So we have seen that a typical angiosperm anther is bilobed. That means it has two lobes and each lobe contains two theca. So what is the meaning of theca? Theca refers to the cavity of the anther lobes. So due to the presence of two theca, we call it to be dithecous. Wherever you see, di or bi written, they always refer to two. Fine. Now, there's a longitudinal groove that runs lengthwise separating the theca. If you look very carefully into the diagram shown here, you see something written as the line of dehiscence. So what is that line of dehiscence? It refers to the line or the region where the anther is going to break open, or rather it's going to split, and the contents of the anther will get released. Right. So if we make a transverse section of the anther, we can clearly understand that it has a bilobed nature, right? So what do we see? That the anther is a tetragonal structure. Why do we say so? Because it is a four-sided structure. Look into this image of the transverse section. What can you find? You can find that in each lobe, there are two microsporangia that are present, right? So each lobe contains two microsporangia That means in total, how many microsporangia are present? A total of 4 microsporangia are present, right? And each of these microsporangia will develop into pollen sacs, right? Then next what happens is these pollen sacs will extend longitudinally. What do we mean by longitudinally? That they're going to extend like this, lengthwise, right? And then they'll extend all through the length of the anther. Whatever is the length of the anther, the pollen sacs will extend all through the length, and they are stacked with pollen grains. Okay, so now let us start knowing about each of these microsporangia, right? If you notice this diagram carefully, which is the transverse section of microsporangia, you can understand that they are, nearly circular in outline, right? And they are surrounded by 4 wall layers. What are the 4 wall layers? First is the epidermis, then comes the endothecium, then comes the middle layers, then comes the innermost tapetum. So I would request all of you to look into the diagram carefully as well as notice what am I writing. So if you refer to the diagram, hear to what am I saying and the writing, all of it will become much more clear to you. Okay. So getting back, the 4 layers are epidermis, endothecium, middle layers and the tapetum. The first 3, their major function is protection. They also have another function. What is it? That is, they'll help in the dehiscence of the anther. That means they'll help the anther to be open and let the pollen grains be released. While the innermost layer that we have, which is the tapetum, its function is to nourish the developing pollen grains, right? So the cells of this tapetum, the tapetal cells, they have very dense cytoplasm and they have more than one nucleus. How so? Because these tapetal cells, they can undergo karyokinesis. That means the division of the nucleus occurs very well, but the subsequent step of cytokinesis does not occur properly. So as a result, what we see is that the one cell contains more than one nucleus. So we can call it to be binucleate. Okay, so the anther, when it is young, it contains a group of compactly arranged homogeneous cells. Homogeneous cells means they are all kind of uniform cells. And these cells are present at the center of each microsporangium. Okay, so these cells are compactly arranged, and this is what we call to be the sporogenous tissue. So now what happens is, as the anther keeps on developing, the cells of the sporogenous tissues, which are originally what? They are diploid, right? They will start undergoing meiosis. And we know that if a diploid cell starts performing meiosis, what will happen? It is going to produce some haploid product. So it forms microspore tetrads, right? Like just to understand properly, say this is a diploid thing and then it undergoes meiosis. So what happens is, first we have N and N in meiosis 1, and then again in meiosis 2, what do we see? That further this N divides into 4 N cells. So what is the ultimate thing happening? That is, from a diploid cell, after the meiosis division, we have 4 haploid cells being produced. Fine. So as each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, what can we say? That each of these is a capable microspore mother cell, right? Or in other words, we can say that each cell acts as a potential pollen mother cell. Do we understand this? So each pollen mother cell will now undergo meiosis, and then they are going to produce the microspore tetrad, as we have just seen, right? So this process where the microspores are being formed from the pollen mother cell by the process of meiosis, this is known as microsporogenesis. So microspore is being formed, so microsporogenesis, right? So the microspores, when they are formed, how are they present as? They are present as a cluster of 4 cells, right? We can say that this is a cluster of 4 cells, hence the term tetrads. Are we clear with this? Now what will happen? As the anther will keep maturing, it will get dehydrated. So now the microspore tetrad, individual components, these 4 cells, they'll start separating from each other. So they'll dissociate from each other, and then what will they do? They'll turn into pollen grains, right? And each of this pollen grain is going to represent the male gametophyte. Is this concept clear? So what have we seen? We have seen that from each microspore mother cell, we are getting 4 pollen grains, right? So what we can say is that inside each microsporangium, several thousands of microspores are formed. That means thousands of pollen grains are formed. So once the anther dehisces, all of these thousands of pollen grains are released. Okay, so these pollen grains, they have amazing architecture, and different pollen grains from different species have various types of structure. So this image, like these 3 images, they show the scanning electron micrographs of various pollen grains, meaning that these images have been taken under an electron microscope, right? So, pollen grains are generally spherical in structure, and diameter wise they are approximately 25 to 50 micrometers. That means you can understand how small they are. Definitely, because they are so small, in order to understand their structures, we have to use an electron microscope, the images of which we've just seen a few minutes back, right? So the pollen grain has a 2-layered wall. The outer wall is the exine, which is hard, and the inner wall is the intine, which is comparatively thin. Okay. Now talking about the exine. So what is it made up of? Yes, it is made up of the sporopollenin. Now the sporopollenin is one of the most resistant organic material, right? So what is it resistant from? It is resistant from very high temperature, means it can tolerate extremes of high temperature. It can tolerate strong acids, and it can also tolerate alkalis. That means bases. So you should be amazed to know this, that till date, there is no known enzyme that can degrade this sporopollen. Okay, so this exine of the pollen grain has very prominent apertures called germ pores. If you notice the diagram very carefully, you'll see that, see, there's an area called the germ pore. So what are these germ pores? These are, gaps from where the pollen tube will come out, right? That is, if the pollen grain goes and sits on its correct species stigma, then what will happen? The pollen tube should come out, which will go and reach the ovule, right? So this germ pore is a gap from where the pollen tube should come out. So the sporopollenin is absent in these regions. And since we have already seen that the sporopollenin is extremely resistant to high temperature, acids, alkalis, etc., what can occur is the pollen grains are well preserved as fossils because of this sporopollenin. Okay, so talking about the inner layer, which is the intine. What is it? It is made up of cellulose and pectin. And this layer, intine, is comparatively thinner than the exine. So we can say that it is a thin and continuous layer. Why do we say that it is a continuous layer? Because in case of exine, we have seen that there are gaps, the germ pores. While in case of intine, there is no such gap. So hence we say that it is a continuous layer. And it is made up of cellulose and pectin. Right? Now, when the pollen grain matures, there are 2 cells present. What are they? One is the vegetative cell and the other is the generative cell. Okay. So if you look into this image very carefully, you'll be able to understand that this vegetative cell is bigger. Why? Because it contains abundant food reserves. And within the vegetative cell, there is the vegetative nucleus, which is irregularly shaped. Now coming to the generative cell, so this is small and it floats in the cytoplasm of the vegetative cell. Okay, and this generative cell, that is the smaller one, this is said to be spindle shaped. Okay, and it has a very dense cytoplasm and nucleus. Okay, so this is known as the two-celled stage, right? That is, one is the vegetative cell, the large one, another is a generative cell, the small one. And 60% angiosperms, or a little more than that also, over 60% angiosperms shed their pollen grains at this two-cell stage. What happens in the other 40% of angiosperms? In the remaining species, the generative cell will further undergo mitosis. So when it undergoes mitosis, what happens? It gives rise to two male gametes. Okay, so now what happens? This is a three-celled stage. That means what are the three cells? There is one one vegetative cell present, and the generative cell has divided into two separate male gametes. So in total, this is a three-cell stage. The rest of the angiosperms will shed their pollen grains at this three-cell stage. Okay, so these pollen grains that we have just studied about, pollens can lead to different kind of allergies, okay, as well as respiratory problems. So we can say that pollen grains can cause bronchial afflictions, right? Especially in people who have respiratory disorders, who have chronic respiratory disorders, like, pollens can lead to bronchitis. What can they lead to? They can lead to diseases such as bronchitis. Then we have asthma, etc. Right. That means pollen grains of several species can cause allergies. Okay. One thing that we have to mention here is about Parthenium or carrot grass, right? So Parthenium or carrot grass, this came to India with wheat. It was imported to India along with wheat, right? What happened was this Parthenium served as an allergen or a contaminant. So it became ubiquitous in occurrence and it caused pollen allergy. So this is just the image of the Parthenium or carrot grass. Okay, so these pollen grains, they also have nutritional value. They are very high in nutrients, right? So nowadays it has become a recent fashion that pollens are being used as food supplements. That is, pollen tablets are being made and they are used as food supplements. Pollen tablets or pollen syrups, etc., are widely available in the market, right? So, you can see this image where you have different kinds of energy shake, pollen creams, etc., etc. Right? It is also being claimed that consumption of pollen can lead to better performance of athletes and horses in races. Okay, so we can say that consumption of pollen gives more energy to athletes and race horses to perform. Fine. So we can say that pollen grains have very good nutritional value. And pollen grains have different viability. What do I mean by this? Say the pollen grain, pollination occurs, the pollen lands on the stigma, right? Now further steps will happen, fertilization will happen. But this time frame is not always fixed, right? So pollens are known to have great viability. Like for example, if you talk about rice and wheat, so these plant pollen grains, they lose viability within 30 minutes of release. On the other hand, if you talk about plants belonging to Rosaceae, Solanaceae, Leguminosae, these plants, their pollen grains maintain viability for even months. Getting it? So what can we do? That pollen grains can be frozen and they can be well stored in liquid nitrogen at minus 196 degrees centigrade. So this kind of storage is what we call to be cryopreservation. Okay, cryopreservation. That is storage of substances in liquid nitrogen at minus 196 degrees centigrade. And these stored pollen grains can be kept in the pollen banks and they can be used for various crop breeding programs. Well, so that's it about the male reproductive part. That is androecium. We've talked in details about the stamen, the pollen grain, what are its functions, what is the structure and all. In the next video, we are going to start discussing about the female reproductive organ, that is the pistil. Please stay tuned. If you like the video, then please like, comment and share it. Also, don't forget to subscribe to my channel by explaining. Let's keep learning. Thank you.